We start on time. It's 4:30, so it's my great pleasure to introduce Eric Holland. Dr. Eric Holland is a director of Human Biology Division at Fred Hutch. He's also a professor of neurological surgery at the University of Washington, and he holds an endowed chair, Alwood Chair in neuro-oncology, as well as a directorship of the Alwood Brain Tumor Center. Dr. Holland came to us a few years ago. He has a very accomplished history in science. He first got his medical degree at Stanford University, and then he went on to do his doctorate in molecular biology at the University of Chicago. He had a privilege of working with two Nobel laureates, Dr. Paul Berg, who pioneered recombinant DNA technology at Stanford, and then he worked with Dr. Harold Varmus, director of National Cancer Institute. His work is focusing on gliomas. He has developed many pioneering technologies in modeling of gliomas in mice, and today he is going to share with us his wisdom. Eric?
Thank you. Okay, so I'm going to talk today about sort of a combination of mouse brain tumors and human brain tumors. And my work historically has been with mouse tumors originally, but since I've come here to Seattle, I've gotten interested in large data sets and big data analysis of gliomas mostly, and the second half of the talk will be about that. Okay, so you all know this, you're neurologists, but this right here on the red arrows is what a glioblastoma looks like. Surgeons usually can take these out, or at least most of it, that you see on a scan, but the tumor almost always comes back. And this is that data. Once you've had pretty good surgery, the overall survival is really terrible. And so on this curve right here, in the red is what happens if you were diagnosed with a GBM, but you don't really do much in the way of surgery, maybe a biopsy only. And in blue is good surgery, nothing else. Green is adding radiation, and in black is adding chemotherapy to that. And then the other curve here is from the Stoop paper. And what you realize is that the problem is that the colored curves here were published back in 1967, and except for the addition of temozolomide with radiation, there's been very little in this space, and you probably know that. Okay, so these tumors, so the question is why don't these tumors respond to therapy very well? And one of the thoughts a few years ago was that maybe it's because there are multiple diseases. There's all with the same word, glioblastoma, and in fact using the TCGA analysis of hundreds of samples looking at gene expression profiling, it fell into subgroups, mesenchymal, classical, proneural, and so on. I'll just tell you, and I'm not going to spend almost any time talking about this topic, but I'm not convinced that these subgroups really matter that much. Although it's not like they're not real, but it turns out they're probably related to each other in a way which doesn't have a lot of therapeutic meaning with the drugs that we have available to us right now. So one of the things that, as I mentioned, mouse modeling is something we've been doing for years. So we can recreate these tumors genetically using viruses. And so we can take the genes that are abnormally expressed in gliomas, and we can convert each one to a virus, and we can check and test whether any given virus, when transferred to the brain of the mouse, where you've transferred that gene, is sufficient to cause tumors. And the vast majority of the genes that are abnormally expressed in human glioblastomas, in fact, don't do anything, but a few actually do cause tumors. And the tumors that they form look an awful lot like glioblastomas. I'll show you in a second. But the system that we use is something called Argos-TBA. And the retrovirus here, called Argos, is a place where you can put your gene of interest in. And that virus, because of the kind of virus it is, will not infect mammals. The receptor for RCAS is not found in the mammalian genome, but you can engineer mice to express the receptor called TPA, and you can then make the cells that express that receptor infectable by the virus. And so you can express that receptor from nestin-expressing cells or GFAP-expressing cells or other cell types. And now you can transfer your gene to a specific cell type. And if that's sufficient to cause cancer, then you get that. And the tumors that you get look, as I said, very similar to human. A lot of pseudopalisade, a lot of microvascular proliferation, invasion into the brain around. And the illustration of the invasion that we see is here. And so this is a mouse that was infected with vectors. One of the nice things about the vector technology is that you can mark every cell that's infected. So if that cell gave rise to a tumor, then all of its progeny are marked, and you can do immunostaining and see where they really are. So in this case, this mouse didn't really have any symptoms, but we sacrificed it in part of the study, and it actually had a small tumor right here. And you can imagine, if it was a little close to the surface, the surgeon go in there and cut it out. But the real problem is that the tumor cells, which if you look under the microscope are clearly here, turns out there are other places too. So if you use immunostaining, you find that the tumor cells are there. And in fact, there's tumor cells that cross the corpus callosum, and so then it's, it looks a lot like the human problem, and that's actually why it's not surgical. Well, let's not say that. It's not surgically curable. There's a value to surgery. It just isn't curing it. Okay, so we can create these tumors. They look like gliomas. They invade like gliomas, and they respond to therapy like human gliomas. And so for example, here's a tumor that was identified in a mouse, and a few weeks later, say 2 weeks, basically it grows to the point that the mouse dies. And you can follow these things with MRI scans. You can radiate the mouse, and we've been playing with different doses of radiation just to understand what they do essentially. And so 10 or 20 gray, either 10 all at once or 20 gray in fractions, and so on. So you can get these tumors to shrink, but eventually they come back and they kill the mouse. In fact, they kill all the mice. And so you can look at survival curves in these systems, and it's pretty. There's a lot of parallels in the human to mouse situation here. So untreated, almost all the mice in this particular experiment were gone in 3 weeks, and temozolomide, bought an extra week or so, and radiation buys maybe 2 or 3 weeks. But again, they all come back and they all die. And we're interested in the biology of the response and the recurrence and so on and so forth. Because they're mice, we can engineer them to allow these kind of studies to be a lot easier to do. And so here we have luciferase transgenic animals in this particular set of photographs where the luciferase is driven by the Glee promoter, which is a readout of the sonic hedgehog pathway. And the upper tumors right here are actually medulloblastomas driven by the sonic hedgehog pathway. So it's not surprising that the tumors in the upper panels are lighting up with this particular reporter. It's sort of given. But the lower panels are in fact gliomas driven by PDGF, which is the strongest oncogene we've found. And probably, parenthetically, the oncogene that actually starts the whole problem in humans of all subtypes. But anyway, this tumor under H&E, lights up strongly with this reporter. And so it turns out sonic hedgehog signaling occurs in PDGF-driven tumors. Exactly how it gets turned on, how it's regulated is not clear, but in the absence, it's really known that. But you can also use these reporters as readouts of therapy. So this is a mouse live in both cases, before and after radiation therapy, and this particular type of tumor that we're looking at here is very sensitive to radiation. So it's one of these medulloblastomas that are p53 wild type. So they have a huge response to therapy really quickly. And so this is just after a few hours, the light just disappears in this tumor. Unfortunately, it comes back in the tumor, and of course, back in kids too. But anyway, we can use these tools. So we can use these tools also to begin to understand, things that we should be doing or shouldn't be doing. And obviously, you could use them to develop drugs, and I'll show you that in a second. But you can also use the models to maybe guess what you are doing and maybe shouldn't be doing. So this is a study that I'm not going to spend a lot of time on, but it probably would interest you a lot. And that is that, as you know, everybody in the OR gets decadron with this disease, essentially everyone. And most people have it going into the OR. And some people are weaned off the decadron and they don't have it on during radiation. And some people, most actually, in most places, have decadron present during radiation. But as it turns out, analysis of the human data very strongly suggests that the patients who are not on steroids during radiation do better. And you could say, well, that's confounded by the fact that they're not sick enough to need steroids, then of course they aren't on steroids. But it's more complicated than that. Turns out that radiation, in fact, the efficacy is inhibited in the presence of steroids. And this just is a figure from that paper illustrating this point. And that is that untreated, just with steroids only, this in the blue is no steroids, and red is with decadron, and the mice go down pretty quickly. Radiation gives this big bump from blue here to blue here, but in the presence of steroids, what you get is in the red. And that's true with fractionated radiation. That's true with one single dose radiation in the mouse. So that's actually caused the people at Sloan Kettering, where this data was originally generated, to actually change their practice, and instead of having about 80% of the patients on steroids during radiation, that number's dropped down to 60. And now the question is, what could you do for the other 60%? Could you do something else like Avastin and so on? So this is an interesting conversation.
Eric, does it matter if they've been briefly treated with steroids?
I don't think so.
Off?
I think it's just a matter of being off. And so, it's a whole hour talk here on that topic, but the steroids actually reduce proliferation. And if you do a gene expression profile of tumors with or without steroids, the biggest differences in expression happen with the cell cycle. And so you're actually, and it may be also affecting the immune system and a bunch of other things, but you could imagine reducing proliferation would reduce efficacy of stuff that kills cells by DNA damage. So anyway, that's out there. It's under review right now, so we'll see what happens. Okay, so the other thing that other things that we can do are, and we do interact with small biotech companies that are developing drugs. And so this is just one quick example of an interesting one, where the drug itself appears to have very little effect on survival. So these two curves are it doesn't even matter what they are, the control versus the drug, and they all go down pretty quickly. But if you actually radiate or in this case, it's temozolomide is the first slide. If you give temozolomide, there's a as I showed you before, there's a median increase in survival from here to about there. But in the presence of this drug, that jumps up dramatically with a significant p-value, and you get the same kind of result with radiation. So radiation works better to begin with, but this drug enhances the radiation effect too. So this is an interesting compound that is under development right now. And there's several other things out there. Some are working with the immune system and other things like that. We hope to get some of these at somewhere into clinical trials if they pan out well. Okay, so but the response to therapy in this case is radiation, so we're interested in the biology of response to therapy. And so if you look carefully at these tumors, they're complicated. They're not just sort of a proliferation of clonal cells sitting in the middle of somebody's brain. They have a lot of different cell types. They're not all tumor. And in fact, in the region of the vasculature, there's a lot of cell types like endothelial cells and microglia and astrocytes and a lot of stroma, if you want to think about it that way. And so this is an H&E picture of that. And one of the things that's in the perivascular space are cells that have stem-like characteristics. Many of them are tumor, but others are not actually tumor. They're just stem-like cells that are non-neoplastic. But you can see them easily by staying with nestin. And I should point out that, although I'm not a big fan of the subtypes of glioblastoma, the proneural subtype has more of this sort of perivascular versus non-perivascular contrast histologically. And some of the other subtypes, the stuff's mixed around more. But the mouse model is a really good model for this particular flavor, essentially. So anyway, the perivascular region has a lot of stem-like cells in it, and the bulk of the tumor expresses a different marker, which is called OLIC2, which is a transcription factor expressed in more differentiated cells. Still pretty undifferentiated, but less than the stem-like cells. But the take-home message on this is that these cells have differential response to radiation. And so if you look untreated and after 72 hours after 10 gray of radiation, which we use a lot, turns out 10 gray all at once and 5 times 2 gray Monday through Friday gives you essentially the same result. And if you're looking at real kinetics, then doing 10 all at once actually can let you see things really nicely. But the point is, three days after that dose, that you preferentially lose the bulk cells, the cells that express OLIC2, and you spare these perivascular regions, which become a higher percentage of the total tumor. And that's, there's maybe many reasons for this. Our lab and others have been interested in, and maybe thinking about this as a stem-like characteristic. So stem cells and stem-like cells are less responsive to DNA damage, and it would be at least consistent to say that they're resistant because of their stem-like characteristics in this region. So we became interested in understanding, coming up with ways of measuring stem-like behavior in these tumors, and there are many markers that you can sort out stem-like cells, cell surface markers, functional markers. We got interested in the site population, which is measured here, just a way of effluxing dye out of sort of non-stem-like cells, and the cells that are more stem-like end up in this part of the flow. You can pull these out; they tend to grow like neurospheres. They express all these stem-like markers, and when you inject them back into a mouse in red here, they kill the mice faster than the other part of the tumor. So, there's a big debate about cancer stem cells, and I don't want to be in that debate. But I don't think there's any question that there's a spectrum of differentiation of tumor cells in a tumor proper, and there are some cells that are less differentiated in that spectrum, and they appear to be more resistant to the standard therapies. They probably are the ones that are giving rise to recurrence, at least preferentially. Okay, so all that's kind of published. We have other models, and so we just came up with a new model for IDH mutant gliomas that did this. It's not fully characterized yet, but it makes the same amount of 2-hydroxyglutarate that the human tumors do, and has some interesting properties that appear to be parallel with the human IDH mutant tumors. We also have developed a model of ependymomas, supratentorial ependymomas. This is human here, but the long story short, there's a fusion gene fusion that occurs in supratentorial ependymomas that are very unique between REL-A and a particular orphan-rated brain that no one's ever heard of before, called C11 ORF95. And that particular gene fusion actually is sufficient to induce these tumors, and essentially without any other background. In this case, they're NK3R deficient, and it looks like that might be contributing, but it doesn't really matter. Wild-type tumors, mice get the tumors just at the same rate. So it's an extremely potent oncogene for this particular tumor type, and it's specific. I'm not showing histology, but it looks like the human supratentorial tumors. Okay, so all that's kind of mouse stuff. We also, by the way, just probably, although this is not quite as far along, have generated a model of ATRT. So if anyone's interested in that, the mechanism for why the genes that we put in cause ATRT, we're still trying to figure out, but it sure looks like an ATRT, reproducibly. Okay, so for mouse work, we use the mouse models to understand, causation, and trying to infer what happens in the human disease. We try to understand the response to therapy, and why certain cell types are resistant and not. And we try to use them to develop and help companies develop drugs that will ultimately be in clinical trials. We did have a couple of trials at Sloan Kettering that were originally piloted in these mice, and they're still ongoing right now. So the pipeline kind of exists. It would be nice to make that pipeline more robust, but that's the kind of ideas that we have. Okay, so that's the end of the first part of this talk, and I'm actually maybe doing pretty well on time. If anyone has any questions about the mouse work before I go on to the big data human stuff. Bruce.
So what terrifies me, and I think a lot of other people that work primarily in rodents, is the possibility that we might be dealing with cells that are very significantly different from the counterparts in the human brain.
Okay.
I'm fairly relieved that that's not the case so much for neurons, but for astrocytes, I'm terribly concerned that it might be the case, which imperils a lot of insights that we gather from rodent work. Do you have an opinion about that?
Okay, so the question, just for those who are listening, is how similar are mice and humans, especially in glia, and if you learn some stuff in mice,
Especially in astrocytes.
especially in astrocytes. Fair enough. So am I worried about that? I mean, I'm worried about everything. You know, if we're not worried, we're not thinking. On the one hand, on the other hand, you know, you study astrocytes per se, and these tumors are pretty not exactly astrocytes. They do express GFAP. They're early on, and you know they're a lot more similar to things much earlier in development. And I don't know exactly about all the physiology, similarities and differences. What I can tell you is the genetics, and there are definitely differences between what we do and you know in the mice and what happens in humans. But they're not. It's complicated. That's what the difference is. And again, I'll just say it without showing you the data was published within last year. The human tumors appear to arise by chromosome non-disjunction. So chromosome 7 is gained several copies, and chromosome 10 is lost in one copy as one of the very earliest events. And then there are additional things that happen. And when you cause cancer that way, it's not one gene. It's like a lot of genes. And there's computational ways of scaling across chromosome 7 and 10, looking for the drivers, and then testing which drivers could actually be sufficient. And it turns out chromosome 7 encodes PDGFA, and chromosome 10, of course, has P10 in it. And so many of the strongest drivers we sort of already know, but there's a lot of other things on chromosome 7 that can contribute. Now, if you look at the tumors that eventually arise in the mice and do a gene expression profile, there are many of the genes on human chromosome 7 are actually overexpressed in the mouse model, even though it's not on the same chromosome. So some of that stuff is selected for you know, once you have a tumor, then you know what's up, right? A lot of these things actually are on, but they may not be on the same way. They're not on by copy number gain. They're on by selecting for the need for them. And so does that affect the therapeutic response? Maybe. But I think we can get a lot of insight about what the questions we should be asking by comparing the two species.
I like that answer. Steve Goldman has a mouse who's been able to ingrain a bunch of humanized neonatal astrocytes. You know that mouse model, I think they published it in Cell Stem Cell. That might be a cool place to investigate this a bit further.
Could be. Be very hard to cause chromosome non-disjunctions in those astrocytes in that mouse, but you know, yeah. Okay. All right. Yes.
What has caused the delay in obvious translational benefit of using mouse models and the practical clinical application?
Great question. I think the problem, you know, to be a complete cynic. For those who are listening, Dr. Chamberlain asks if we can do stuff in mice, why can't we fix it in people? And the truth is, if you have a really good mouse model, those mouse models respond to therapy just exactly what humans do, which means they respond and then they recur and they all die. That's not the case with many xenografts and other things that people have actually developed drugs based on. But the truth is, you know, we don't have anything good for humans. We just don't really. And you know, if you had a really good mouse model that really mimicked that, that stuff wouldn't work in the mouse ultimately either, and they don't. So the question is, you know, what's next? What, you know, because the standard things like the inhibitors of EGFR and you know some of these other tyrosine kinase receptors that sounded like they might be good ideas, because those mutations were found in the human disease, they didn't work. They don't work in the mouse. They don't work in people. so the idea that those are critical components of this disease appears not to be the case as far as a therapeutic target. Now, you can cause a disease with gene X, but that doesn't mean that once you have an established disease, you attack gene X, that's actually going to be sufficient to turn the whole thing around. In CML, that's probably the case with, a specific gene fusion where the rest of the chromosome or the genome is stable, and there's a single event that takes that cell and makes it neoplastic. And if there's a drug to that one thing, you get the Gleevec, effect, right? And that may actually be true in the case of these ependymoma, where appears to be a single thing. I don't know what the drug would be, but that would be a great target if you could come up with one. But a real glioma is, it's like Dresden after the war. I mean, it's a mess, and you can't say that if you just put this brick back together, you're going to fix the whole thing. It's not the case. So, we use the mouse models to try to understand what to do, but expecting there to be a silver bullet coming out of them, I think is expecting too much.
They did put Dresden back together.
They did, and it took a lot of work.
But it's doable.
And it's doable.
They put what was left of Dresden back together.
Right, and then they came up with some, new blocks. Yeah, but you're right. You're absolutely right. It's doable if you have the blueprint, but you got to learn the blueprint, right? Okay, so the second half of the talk is big data. So this is a project which was started at the Hutch, but it's part of the Hutch, the SCCA, and the UW, and it is piping all of the clinical data from the medical record, from study databases, and from all sorts of other places into one central repository that will ultimately have molecular data associated with it. So you can actually use that big database and learn something. Okay, it's a lot of words. It's called Hydra, and it's a multi-million-dollar effort which started several years ago, and it's beginning to wind down. Right now, Hydra, the pipes finally got turned on just for the basic stuff a month or two ago, and the total number of patients that are basically UW patients that are all cancer, starting about 2000 until now is about 335,000. They're not all fully treated here. Some of them are simply coming here for second opinions, and some of the stuff will be really minimal. And many of the patients who actually were treated here don't have very good clinical data. I mean, this is a real problem because we don't follow the patients very well here. But some of them were followed. In order to do the kinds of stuff that I'm going to show you, you really need a database where from diagnosis to death, you have all this information, you have the survival, you got the data recurrence, and all the rest of it. If you have all that, then that's useful data. If you don't have all that, it's not that useful. So that huge number is full of not that useful data, but there is some good stuff in it. And that's across all cancers. That's not just obviously brain. So this is sort of an idea that not only does it have assays and subjects and studies, it has specimens, it has all these other things. And so how do you pipe in the medical record? The medical record is, some of it is just numbers, so that's easy to pipe in. A lot of it is free text. And so, you have to actually pay people to read through it and extract the data out, or you have to have, using natural language processing algorithms, extract the data out and verify that you're not wrong about it, audits and stuff to make sure you're right. This is a big effort that's ongoing right now to try to fill in this initial scaffolding of this thing called Hydra. It has, excuse me, it has all sorts of issues. You're putting in data from multiple institutions that have to agree legally and privacy-wise to mix it all together into a place in the middle here that has a strong privacy shell around the outside of it. And then who's allowed to reach in and pull that data out again? You have to be able to, sign off that you're, the right person, and you're doing it for the right reasons, and you either need patient-identified data or de-identified data, and so on and so forth. So these agreements have taken a huge amount of time. I've not done them. I just want you to know that there's a lot of people working on this to try to make this work, and it's only for 3 institutions. Imagine what it's going to take to make it work nationally, but nonetheless, that's obviously where it needs to go. But Hydra itself is just a network of data. you need to have a front end, a interface where clinicians and researchers can actually begin extracting data out. And so this is a separate entity called Argos, and I forget what it stands for, something. But this is the front end. So I'm going to show you, and Argos isn't quite up to speed. It's getting there very, very soon. I'll show you a little bit of what Argos is going to look like for you guys, for anyone in cancer. You're going to be able to play around. But Argos for brain is one of the first ones that have come up. And so there's a log. First of all, before you even get going, you'll need to take the right HIPAA training. You're going to have to verify that you are who you are, and all these various things that allows you to get in. You'll sign in. You'll pick an organ site. Right now we've got a lot on brain, head, neck, and lung, so you'll pick one of them. You got to choose the activity. Is this research preparatory to some sort of a trial? Is it just flat out research, de-identified data and so on? Got to verify a bunch of things, and then what's going to come up is what does the database look like? And so I'm just going to this is a work in progress, and so we're adding more and more stuff to Argos right now, just to be useful. And I'll say also that the really interesting stuff is after Argos. So just as you're watching and you're thinking it's not that interesting, just wait. But this is where all the data is. This is where you can go into the data and say, well, how many patients do I have that are that have this particular disease or had this chemotherapy, or there were females between the age of 25 and 35 back in the 2007 that whatever, all that stuff is easily queryable and is spit out by this program. You can figure out what kind of radiation they had and narrow it down to subtypes and so on and so forth. You can even if you were going to run a trial and you say I'm going to run a trial and it's going to be for females that have a particular tumor type which might be kind of rare and it's got to be certain ages, whatever, you can mock that up in the system and say, what is the accrual rate going to look like and how long will this actually take? And it turns out it'll take 17 years or whatever. I'm not going to run that trial, right? But this, a lot of times we write trials that we don't really think through what it's really going to take, but this will actually tell you instantly whether it's a good idea or not. So we're filling all this in. There's a lot more stuff that's going to go into Argos, as you'll see in a second. But the brain was the first, and then we've got head, neck, and thoracic, and so on. Just moving them out. Okay. We're heading for a March 15th rollout for Argos, and some of the diseases are actually on board a little bit sooner, and brain is one of them. And so people are really interested in this. One of the issues that we still haven't gotten over from the right standpoint is that people at the UW, because it is sitting at the Hutch, you got to have the right permission to get in, to reach through into the Hutch's databases to do this. And so we're working on that. But clearly, the U is going to have to get in, otherwise it's not any value. Okay. But the real issue is, Argos is kind of clunky at this moment. It's interesting data, but it's not the fun data, right? Fun data is this other sort of playground we call Aquascape. And Aquascape is a way of visualizing data so you can really understand it. And I'm going to run you through this in a second, and I think you'll understand what I'm trying to say. But we are developing things in Aquascape that will be rewritten and put into Argos so that you can just, in the Argos format, which is very stable and isn't going to crash, and so on. So it's really useful to clinicians to understand this disease. It'll end up in Argos. Okay. So the idea with Argos, with Aquascape, is that it's this multi-dimensional matrix that you can pull out the subsets and look at interactions between them. And the people involved are Lisa McFerrin, Paul Shannon, Hamid Bellari, and Jenny Zhang. And I'm sort of the clinician that sort of, as they develop these things, I sort of tell them what I think it should look like. Oops. Okay, so where is... I had it up.
So it's on the screen.
Oh, it's on the screen. No, it's not here. All right. We're sharing. We're sharing, but not with me. Okay. All right. So So this, all right, so this is what Aquascape looks like. And it's under development right now. It's basically a series of tabs, and you choose the tab that you're going to use to interrogate the data. And the data that I'm going to show you, we're building this tool based on TCGA data right now. And the reason we're doing that is because TCGA data has, it's not great, but it has at least reasonable amount of clinical data and a lot of molecular data. And it's a tool, and so once we get the UW data up at the same speed, we're going to pump the UW data into this, and then you're going to be doing this with your patients, right? But it's a tool that's being built right now. And I guess the other thing to say as we move along is that we are collaborating with people outside, and so we are envisioning each of these tabs being written by different people for different purposes, and it's going to be like iPhone tabs, where people, or iPhone apps, are going to write the app that you want for your analysis, and you're going to put it by WebSockets right into this, and we have people who are gearing up to do that. All right, so this, for example, is a simple thing. It's a clinical data. It just simply, it's the table, or and you could, and this has hundreds of patients in it. It's not that interesting. However, if it has all the patients in it, but if you later on identify a subset of tumors that you want to study, you can move all that into this tab, and you'll get all the information on only those patients. So timelines is not work very well. I'll do the best I can, but this is not working the way I'd like it to. If you imagine each patient is a horizontal line, and this is the dates they were born, and horizontally you end up, all the bad stuff happens out at the end here. You would say, this is, it's kind of chaos, but that's what it all looks like. But you can align. You move things around in this thing, so we can align it by diagnosis. And so now the brown line is everyone's aligned up the day of their operation when they were given the diagnosis of GBM. And then as time goes on here, now we're mousing over different tumors and you're picking up which ones they are, but this purple color is radiation, and chemotherapy is in blue after that, and then other things happen after that. So you're beginning to see clarity, but in fact, you see a lot more clarity if it works. You order it by survival, and now what you essentially see is a Kaplan-Meier plot of, you see two things. The top are people who aren't yet dead in the TCGA, but the bottom are all the ones who have passed away. And you can basically, you're looking at, this is glioblastoma. This is what happens to you. You will fall somewhere on this plot. You're one of those lines. It's all the stuff that happens, radiation, chemotherapy, and so on and so forth. This is interactive. You can identify subsets of tumors that are particularly interesting. You can take that, and you can, for example, move that back into the clinical data table, or you can analyze those tumors in other ways and compare them to other things. So anyway, that's a very cool plot. PCA is principal component analysis. Each of those dots is a different tumor, which you can mouse over and see which tumor it is, for example, that one. And the colors are simply whether they're mesenchymal, neuronal, and so on. But the location on that has to do with, there's ones that are very close have similar expression patterns to each other. And that's basically how they got defined in this the subtypes again. But you can grab a hold of a series of those tumors themselves and analyze them as well. It's totally interactive. You can move those back to timelines. You can move them back to clinical tables and so on. This is an interesting tab. So this is all happening on the fly, obviously. So this is it's on Google Chrome. You can log in and play with it yourself if you want. So this is an interesting one. This one is called PLSR. And so what this is once we calculate it, anyway, it's thinking too much. But what this would show you, if something happened, I'm worried about it, is that there are a bunch of dots here, and the location of those dots are genes, and their location has to do with axes of age, of diagnosis, and survival. So you can identify genes associated with with particular outcomes. So that's actually a way of blending clinical and molecular data together and identifying things. Okay, so this is one which is sort of interesting, and so what this is in this space right here in the middle. These are all the genes that are differentially or recurrently mutated, amplified, or deleted in the TCGA above a particular threshold. And I have organized them in, I can zoom in here a little bit, by chromosome. These are the genes on chromosome 12, and X, and 11, and so on and so forth. The size of the gene, the size of the dot there, has to do with how often that is mutated in tumors. The dots around the outside here are the tumors in the TCGA, and the colors have to do with the subtypes they are. Everything is movable, and so it's like a workbench to play around. You can select individual. You can select individual tumors, or a couple of them, and apologize for all this. This is not. The resolution of the screen is not what I got messed up here somehow. This is too bad. This has gotten all screwed up. But I'll just tell you, because I don't know how to reformat the size here. But you can connect individual tumors to all the mutations. You can identify individual mutations and connect them to all the tumors. You can select the tumors that have a particular mutation and take them and send them to a Kaplan-Meier plot that asks the question: is that mutation associated with worse or better prognosis? You can identify individual subsets of tumors like that, and then you can ask for all those tumors collectively, what are the mutations that are found in them? And it turns out the classical tumors are frequently, recurrently having deletions in this group of genes on chromosome 9, which include inquiry an amplification right up here on chromosome 7, which is EGF receptor. But and it's totally interactive. You can grab hold of the genes and move them around and see all the connections move. And unfortunately, it's not doing what I want it to do. But it's a work in progress, and each of these tabs is we're adding to the tabs all the time. Just put it that way. So this is TCGA. So this data is publicly available, when we ultimately do this at UW, each patient is going to have gene chip type analysis, and it will all reside in a very big database where each one of the patient's molecular analysis will be affixed to their clinical data. And it simply pipe it all through this tool, which is the wrong format here. Okay, so
Eric, the data would come from a biopsy.
it probably, at the end of the day, it's probably going to come from paraffin sections of surgical samples. I think that's what's really going to happen. That may not be happening. I mean, right now, much of the data is coming from frozen samples out of the OR. But the technology is evolving rapidly so that we can actually get this coming out of paraffin sections.
Stuff that was in a bank someplace, but not properly.
Right. And stuff associated with people who've already lived long enough to have died. So they actually you can actually make a decent reference database. And ultimately, what you'd like to do is to say, here's this reference database. We have hundreds of patients that we know their full outcome, what they responded to and didn't, and so on, and their molecular information. And now here's my new patient with the same molecular analysis. And how do I use this reference database to tell me what's going to happen with this patient, what we should do with it?
And just thinking as a novice with regard to this patient population, it strikes me that these tumors are unstable enough, some of them anyway, the angry, aggressive ones, that it would be valuable to have an end sample, near the time of death, to look at the tumor, to see whether it had drifted, changed,
Sure.
Substantially altered, and that's part of its capacity to resist all the treatments.
Yep. And that would be autopsy samples or something like this. We don't do that much anymore. But yeah.
That's the problem.
Yeah.
So Barry, the TCGA group has been very interested in
Mhm.
using big data. I'm sure of doing sort of mimicry systems. How does this differ?
Right.
What's unique about
So we work with CBioPortal people and TCGA a lot, and everyone's got their own sort of flavor. This particular one they're really interested in because all the other systems like the CBioPortal has been written as a sort of monolith. It's this one thing, and it does what it does, and it's very hard to add additional functionality to it. But because of the way we've created this as plugins, we're going to basically crowdsource to have people simply continue to add things to it, and it's going to be like your iPhone, where you're just going to let people do what they want to do. And at the end of the day, the people who take care of the patients and really study that particular disease site are the ones who are going to want to know what applications they're going to want. They're going to be much better at making decisions than some group in Boston who decides what everybody wants. Okay, so the last little bit here is this, and so this is not yet an Aquascape. It's going to go into Aquascape, shortly, but right now it's at the pre-Aquascape stage. And so the idea here is that you can, similar to what I showed you for gene expression profiles and PCA plots, where you sort of the location of any given tumor in 2 dimensions has to do with the similarity to its neighbors. You can take very complex data sets like methylation patterns across 450,000 markers and so on, and collapse them down into 2 dimensions. Or you can take subsets of methylation patterns, and collapse them down. And so what Hamid Bellari has done is to start playing around with this concept of multidimensional scaling analysis, which is a little bit like principal component analysis, but it does that. So you take the set of information you want across the entire TCGA or large numbers of patients, and you simply collapse it down into 2-dimensional space. So in this case, every dot here is a tumor, and all 9 panels have the same tumors on them. This is a combination of GBM and low-grade glioma TCGA data. But each panel is a different subset of data that has been used to create this, what looks like a Rorschach test. This top corner right here, the probes used here were the 1,500 methylation probes that were used to identify or define the SIMP status in tumors. And I'll show you what's SIMP and what's not SIMP in a second. This, for example, is methylation in the promoter across all the genes in the genome. This is methylation within the gene. This is a methylation of the 5,000 most differentially methylated probes across the genome within GBM, and so on. And some of these others have to do with copy number alterations and significant nucleotide alterations and so on. So these are just simply ways of collapsing everything down. The specific location of every dot is very specific, as you'll see. And for example, you can color them just to know what's going on. So if you color this group of genes, of tumors, blue in this particular spot, those same tumors are located over here in this one, and over here, and here, and here. And so it's the same ones, but it's like you're looking at a 3-dimensional structure or the shadow of one, and you rotate it and you see things slightly differently, but it's always the same ultimate tumors in the spot. So the question is, let's just start out by asking the question: where is SIMP and where is not SIMP in this plot? So one way to know what SIMP and not SIMP is and isn't is to ask where the 1p19q deleted gliomas are found. And it turns out that the 1p19 gliomas are found right here, and they're not found here. And so this line here essentially divides the SIMP from the non-SIMP tumors, which is fine. But interestingly enough, if you go over and look at the methylation pattern of the within the promoters or within the genes, you still get interesting clustering and the utility of that data to put the these 1p19q deleted tumors into specific places. And not surprisingly, in the case of copy number alterations, these 1p19q deleted tumors here are very centered in one place and very different than everything else and across the entire spectrum. So just to verify this, the IDH mutant tumors are also, not surprisingly, found here, so where you would expect them to be found, and you can also track them across all of these ways of looking at the data. So this is, again, this is where the SIMP tumors, in this case, specifically IDH1 mutant tumors, live. For those of you who think about this a lot, I was a little surprised by the fact that they're basically this whole collection are SIMP tumors, and yet it's a relatively smaller than I would guess, number of those tumors that are actually IDH mutant. So we often think of IDH mutation and SIMP as the same thing. There's a lot more SIMP-like behavior out there than just IDH mutation. That's one thing to know. So another thing that's sort of useful is to then color these things in by grade. And so here blue is GBM, green is grade 3, and red is grade 2. And what you can see, we'll just start here again, is that you have the Seahawks colors and you've got Christmas over here. And what does that really mean? What it means is that the GBMs tend to live over here and mixed in with some of the grade 3 tumors. And there's another good set of grade 3 tumors that live over here mixed in with the grade 2 tumors. But you get a really nice separation of 4 and 2. But as it turns out, the 3s that live with the GBMs have a shorter survival than those 3s that live with the grade 2s, which I guess is what you might expect. But then you can keep on following it down. Certain areas are GBM neighborhoods and bad neighborhoods and good neighborhoods to be in. And what you can always imagine is, once this is all established, your patient with all this analysis is going to end up somewhere, maybe there, and then on this there, and then over here, there, and so on and so forth. And you'll be able to tell a lot about what that patient is by just seeing where, what other tumors are living with it in the various ways of looking at the data. We have ways of coloring in the survival, and it turns, without showing you lots of data on it, this is the GBM neighborhood, so not surprisingly, they have very short survivals. And this is the area where most of the patients are actually still alive, and those that died live a very long time. You can tell that by the coloring. And one of the things that, just to point out here, is that you probably didn't notice this. I'll just go back up. And, if you look at this group here, it's like a doublet. And almost no matter what you looked at, it's about 50-50. And there was nothing different about it as far as we could tell. And so, survival and, grade, and it's all the same. So what is that? And so we thought maybe, maybe there's so this is a group of 5,000 most differentially methylated genes across the entire, or probes across the genome, and it's very likely that some of those probes are actually on the X or Y chromosome. And so if that's the case, then you might guess that these actually might be representing gender. And sure enough, if you color them by gender, that's exactly what they are. And also, as it turns out, although the gender is completely mixed everywhere else, this one is also separated out by gender. And this turns out to be 4 genes that are associated with a migration from proneural to mesenchymal character, at least in the literature. And it turns out that one of them is on the X chromosome. And so that's actually why you're getting that split there as well, sort of out of interest. So, you can sort of, and there's a few more slides at the end, and we're basically finished. But I'm trying to illustrate the fact that there's a lot of sort of interesting data across all of this whole disease, that you can just look and scroll through. And I've learned more about this disease by looking at these plots in the last 2 weeks than I think I've learned before that. Because you're not studying one thing, you're not looking up a paper in PubMed or whatever. you see the whole thing, and you see what's similar, and what goes with it, and so forth. EGFR amplification, it always lives over here for the most part, not over here. Chromosome 10 is lost in several places, but when it's concurrent with EGFR amplification, it's a GBM, essentially. What else could I tell you really quickly? So P53 mutations, I'll skip that, it's complicated. But anyway, P53 mutations are very commonly found which are, in this case, in green, I believe, my eyes are correct, are living over here. And they're not really found in the GBM so much. And I guess we sort of knew that. And they're not found with EGF receptor amplification. We sort of knew that too. But you can now look at other regions here. And it turns out that the lower grade tumors are really divided into two groups. This is a sort of take-home message. There are the 1p19q deleted tumors that live in this area and down here in this plot. And then there's the tumors that have mutations in p53 and several other things that are recurrent. And this makes up a completely separate group of lower grade tumors that turn out to have a worse outcome for lower grade tumors. RB tends to be, again, found in this particular very specific region that I just got done telling you. When it's mixed in here, it's mixing with all the low grades, but these other ways of looking at it pulls apart the subtypes into various groups. There's a mutation on chromosome 11 that we never even sort of knew about, but we were playing on Aquascape, and this thing showed up in the GBM data. And it's like, that's interesting because you could see it visually. And then we took it over down to the lower grade tumors, and it turns out in the blue here, it separates completely from the 1p19q deleted tumors, and it's essentially a low grade marker, and it's an absolute. There's no overlap between 1p19q and chromosome 11 deletion. So that's, again, it's another way of really showing it's two groups.
Where would ETRX fall?
That's a great question. I don't know. That's one thing we need to add to this, right? And that would be really interesting to. We have to do that because that's what people are using right now. This is like more data than anyone really uses, but at a minimum, we need to put the few things that we actually commonly use. Yeah. Anyway, so that's it. I think this kind of analysis is going to be on Aquascape very soon. And once it is, you can just, you can learn about this disease pretty quickly. And people outside are developing, statistical tools and other things to plug into this, and we hope to crowdsource it out to, have everybody use it, and take the UW data and pump it in, so you can actually look at your patients. So that's it.
Thank you very much. Don't disconnect yet. We'd like to ask a few questions.
Okay.
So maybe just a couple of thoughts looking at the last bit of data that you were showing us. It seems to me a little along the lines of the point I was making before. Some of your patients will undergo a couple of neurosurgical procedures.
Yep.
They may have a recurrence, and debulking might be rational. It could be very interesting to look at first biopsy, some treatments,
Yep.
and a second biopsy.
Absolutely. So Dr. Ransom is pointing out that if we have multiple biopsies saved and analyzed this way, that you could actually watch, a tumor that started out over here and eventually migrates up here, and you could actually see a vector of change over time. And that would actually give you some information about, if the surgery was 10 years ago and then at recurrence, it's a much different tumor. You know that the original location that it was found in had a 10-year survival, but now it's not in a neighborhood that has a 1-year survival, and you kind of know. We sort of know this already, but it helps define the neighborhood's meaning, essentially.
And ultimately, you hope it might cough up a secret about what is the troubling migration of
Right.
genetic change.
Right. I should tell you. I'll tell you right now that the tumors in this very region here, which can be found in other places on these other plots, are relatively low grade. They live a long time, but as these tumors migrate out away from this, sorry about that, into this region, they have a very short survival. And all of the SIMP GBMs that were the secondary GBMs, they all live in this sort of in the foothills of this thing. And so the migration, if you're a SIMP tumor and you become high grade, you don't ever move from here to here, because that's actually a different disease. You move from here down here, and this is bad, and this is good. So I think what would actually happen in your scenario is that you would start here, and then you would migrate down here, and then you would die.
That would be interesting, but what I'm hoping for is something that's miraculous, that you discover by looking at a huge amount of this data, that there's a change that you think you could affect, because it just, you know, suddenly all these things kind of come together.
So Dr. Ransom would like a miracle. Okay.
So the challenge you see in the molecular data, clearly we have a lot of this. You can stratify patients diagnostically. Most of it has no predictive value. And even more worse, it doesn't impact treatment. They still get the same treatment. Now you just tell them you're going to die early or later. But I can't do anything about it.
Right.
And that really is the rub.
Well, it's the rub in the clinic, but it's not that different than it was before. I mean, before you had 4 grades. You grade 4, I can't do anything. At grade 2, I'll wait, and then I can't do anything. I mean, the truth is, you know, it was always that way. And if you could actually be better at telling somebody how they're going to do it, it's at least better than it was. But I totally agree with you. What you'd like to do is actually have a binary decision. You're in this neighborhood, therefore you get that therapy. And you're in that neighborhood, you get a different therapy, right? The problem is you don't have that many therapies to do anything at this point. Now, if we start doing trials and have all that data actually in plots like this, I mean, there are some people that are rare responders or that respond to some degree, right? Are they in the right neighborhood for that particular drug to actually give you a good response? You know, you need to populate this with actual data at this point.
But there's been long-term survival data on high-grade surprisingly, when we do sort of retrospective molecular analysis, it's not that we can really anticipate long-term survival. So, you know, Michael Weller has done some nice work with long-term survival for glioblastoma, and shown that most of those were unmethylated. Quite surprising that isn't. But otherwise, we differentiate.
Well, right, but they didn't have a lot of data. I mean,
Correct.
I mean, they're looking at one gene, right? I mean, if you look at enough data, enough granularity, you're either going to find something or you're not. But in the absence of enough granularity, it's not surprising you don't find much. I mean, that's the truth. It's like watching TV with 2 pixels. I mean,
One more question to return to a point that you addressed a bit earlier. If we've got all this opportunity in mice, and I'm just trying to reconcile what we know about the human, what we know about the mouse. I thought I heard you say that up to this point, we largely have seen very good correspondence. If it doesn't work in the mouse, it's not going to work in the human. If it works briefly in the mouse, it'll work briefly in the human. Is there a single instance where there's a complete lack of reconciled data where the mouse behaves in a particular way, it's been tried in the human, no.
Okay, so I don't know if everybody got all that, but the question is, so mouse human again. So it depends on the model you're talking about. You know, historically, mouse models have been xenografts in drug development. And people have cured xenografts over and over again and taken the drugs to the clinic and they don't do anything. So there's a huge disconnect there. But pharmaceutical companies and many researchers just don't really care about that because it's easier to do that, right? If you forget the xenografts for a second and go to the models that actually have the right histology, at least, and the right blood-brain barrier and right immune system and everything else, then the standard of care seems to respond the same in mouse and human. And there was somewhat disconnect, I'll tell you, an anecdotal disconnect. And that is that in the mouse model that's driven by PDGF, if you have a wild-type background, the only thing that's wrong with it is PDGF drives the tumor. If you use a PDGF receptor inhibitor, you can actually get that tumor to settle down, largely. You take that same PDGF receptor inhibitor, you put it in humans, it doesn't work. It turns out, at least one explanation for that is that PGF alone never happens, right? So there's other stuff there. And the most common other thing in those tumors is loss of INK4ARF as a tumor suppressor. If you delete INK4ARF in the mouse model, that drug no longer works. And it no longer works because the tumors, it's almost like in the absence of INK4ARF, the PDGF signaling is amplified to the point that you're not going to actually shut it off. It has an amplifying effect on the signaling that drives it. And so a small molecule is just never really going to pull it off, and it doesn't in the mouse. And so, is that parallel? Is that not parallel? It's an explanation for an apparent disconnect. But what's not been the case, well, there's not that many people using these kind of models in trials because it's a lot of work. And so, I don't know yet. We have some drugs that look pretty interesting right now, and if we get them into trials, we'll see whether they have any effect or not.
So just to do another thing with the mouse is that Bruce mentioned briefly there's a difference in the stroma. Let's just skip the Aquascape for a second. The stroma is honestly quite different from mouse than it is in human. A third of the tumor is constituted by macrophages.
What makes you say it's different in mouse and human? It's actually not that different. It's the same.
We're just talking about the delta. Is that relevant?
Well,
Obviously, mice are not humans.
well, okay.
Yeah.
In most cases, that's true. But what I can tell you is that the stroma is surprisingly similar. The type, the cell types, the location of them, it's very similar. The response to radiation of the stroma, it's really similar. That doesn't mean it's identical. It will never be identical. But the stroma is probably more similar than the tumor cells themselves. The tumor cells have all these big chromosomal problems in humans that don't happen really in the mouse. But the stroma doesn't have that. The stroma is just reacting. And the stroma is, largely the percentage of microglia between human and mouse is like the same. The location of those CD68 positive cells or IVO1, whatever, is the same. The histology, the pseudopalisades, they're indistinguishable. I mean, they're really similar. That doesn't mean they're the same. They're not the same, right? They're not the same in all sorts. The genes, the order of genes on the chromosomes are different, right? The chromosomes have been shuffled between mouse and human, right? So chromosome 17 in the mouse is not the same as chromosome 17 in the human, and it's not even contiguous necessarily, right? So if you're talking about big chromosome sections, that's the biggest difference. Because humans achieve the goal that way, and the mouse models aren't achieve the goal that way because they're not even contiguous in the mouse. So you can't amplify up, random bits of the mouse genome to mimic the gain of chromosome 7, for example. It's not going to happen. Makes sense?
Eric, this has been absolutely terrific, and it strikes me that we should have a megaphone to broadcast to the world in the way that Swedish has been doing. We are preparing a kind of personalized approach to managing brain cancer and other cancers that ought to provide in the fullness of time some answers. So thank you so much.